Coming up next on episode 16 of the Bill Gephardt Show.
Skycam maneuver has started. About 20 meters off the surface.
We're getting signals from MRO.
Touchdown confirmed. Perseverance safely on the surface of Mars, ready to begin seeking the sands of past life.
NASA's Perseverance rover lands on Mars and the search for ancient life form begins. And guess what? That search for ancient life begins right at the doorstep of a Utah professor. Hear how her studies of the Great Salt Lake get her involved in playing a role in the discovery of life, if any, on Mars.
The question is, if there ever was life on Mars, was it salty life, and is it still buried there in the salt?
This podcast is brought to you by these proud sponsors of local journalism. Robert J. DeBryan Associates. When you're injured, your attorney matters. All Utah Plumbing, Heating, and Air. Let John Holland and his team keep your home safe and warm this winter season. One Wire Fiber, where Adam Sessions and his folks help businesses communicate better from the office, home office, or from the road. And by ES Solar. Save with solar power today, the clean, safe, affordable energy of the future. And by our special guest sponsor, Legacy RV Center. Well, let me welcome to the podcast now a face you might not recognize, but you will soon enough, is my bet. It's Dr. Bonnie Baxter. She is the chair of the biology department at Westminster College. She's a biologist, and she's the director of the Great Salt Lake Institute at Westminster College. Welcome, Bonnie.
Glad to be here.
But the reason we have you here is because of the landing of the Perseverance on Mars and your involvement with that. What is, just as an overview, what's your involvement in that?
Well, I study things that live in Great Salt Lake, and where Perseverance landed in Jezero Crater is actually a former lake. And as that water was drying up on the surface of Mars, that lake would have become salty. And so I'm really interested in the salts that were left behind when the water went away. And that's a bit of what we've seen here with Lake Bonneville evaporating since the last ice age and forming this puddle at the bottom that is Great Salt Lake.
13,000 years ago or something?
Yes. Yeah.
Okay.
From 15 to 13,000 years ago, the formation of Great Salt Lake. So around Great Salt Lake is a lot of salt. As the water dries up, the salt that is around there holds on to these microbes over time. So we've been collecting salt, and 10 years later, we can grow microorganisms out of them. And we've actually done some experiments with ancient salt where we can grow microorganisms. And so the question is, if there ever was life on Mars, was it salty life, and is it still buried there in the salt?
Let me go back. You grew microorganisms out of the DNA you harvested from inside of salt crystals?
Yes. So lots of people that study salt biology have worked in ancient formations. I've been under Carlsbad is a Permian salt formation that's older than the dinosaurs. It's 250 million years old. So I have worked there.
So you went down into the mine?
Yes. Yes.
And harvested salt from in that mine?
Yes. And inside the salt are these tiny little fluid pockets. And that fluid is 253 million years old. And inside that, you can find remnants of the microbes that used to live there. And sometimes you can find the microbes themselves.
Okay. So let's separate what might be found on Mars and what you have found. For those of us who've seen the movie Jurassic Park, that involved amber and a mosquito getting caught inside of the amber. And then they reproduced dinosaurs, from the DNA that was found there. Are we talking about the same concept except in salt?
Yes. And that
yes, qualified, yes.
Yes, asterisk. That amber experiment was based on actual science, where mosquitoes were trapped in amber from dinosaur periods, from Jurassic periods. The idea that you could take DNA and piece it together and make an animal is a little far-fetched.
Okay. But
you could certainly sequence DNA and understand how how that DNA is related to things that are alive today. That's certainly possible. So a little bit sci-fi, but a little bit real. And yeah, that's a great analogy. It's exactly the same. Little time capsules of biological material in the rock record. And why wouldn't that happen on Mars if life had been there, right? Because the laws of geology and physics, it's all the same, right?
Yes, yes.
Yeah.
So is the rover Perseverance going to be able to segment out the DNA and recreate Martians or allow the potential? How's that work?
Well, so first of all, we don't know if life was ever there. One question that Perseverance will answer is the planet, I'm going to quote a friend of mine, John Armstrong, who runs the Ott Planetarium at Weber. Is the planet habitable is one question. That doesn't mean it was inhabited. So the first question is, was it hospitable to life in the first place at the time that Earth was beginning the formation of life? And then the second question is, well, was there life there? So we don't know the answer to that. If there was life there, so that's the big if,
Yep.
But if there was life there, then we get to your question, which is, did it leave DNA behind, or is it still there? Can we find it? But there's a sub question underneath yours, which is, if it's a different origin of life, would it necessarily be DNA-based life? Like all our life on Earth is related to one common ancestor that had DNA as genetic material. So all life on Earth has DNA as genetic material. If there's a different kind of life as we do not know it on Mars, then we can't assume it has DNA. But since that's the only way we understand life, it's certainly something we're looking for.
Have there not been experiments or findings of some sort that show in the ammonia ooze that started Earth and the solar system, indeed, that have they not found other compounds that might imitate DNA?
Yeah, no, that's there's a huge field of study called abiogenesis, looking at when we went from abiotic, no life, to biology. And that's separate from evolution. Evolution is, once you get biology, how does it change over time to produce all of these species? So the idea that you're pointing to, that leap from chemistry to biology, is something that you can recreate pieces of that. And so there are people that work on recreating nucleotides that make up DNA or RNA.
Okay.
And they're doing that with what would have been on primordial Earth, the chemistry that would have been on primordial Earth. So also, so that's definitely, there are people doing experiments with that right now.
Okay, so it could be DNA, it could be something else, but whatever it is, we're trying to find it. What is the Perseverance going to do for you, Dr. Bonnie Baxter?
Oh boy. I'm getting excited about sample return. So that won't be for a number of years, probably like 6 years.
What are they going to do? Just fly it back? I thought it was a one-way trip, but is they going to fly it back?
Okay, this is the coolest thing. So it's a one-way trip for Perseverance, but Perseverance has a friend who's going in a couple years called a fetch rover. And that fetch rover comes down in a vehicle, hops out of it, and goes and picks up all the samples that Perseverance has carefully dug. Perseverance only has the ability to dig like 40 samples. So it's going to choose very carefully where it digs. The fetch rover will go grab all of those samples and return them to the home base of the fetch rover, which has a rocket. It's going to load the samples in the rocket. The rocket goes into orbit with the samples. Now we send another vehicle into orbit because that's easier than a lander.
It all sounds like a lot like work to me, but go ahead.
Isn't it fun?
Yeah.
And then that orbiter meets up with the sample rocket. The samples get loaded and it gets brought back here to Earth and delivers the samples. Guess where?
No kidding. To you at Westminster College?
No, to Dugway.
Oh. Well, it was a good guess.
They're going to be plopped down in the West Desert of Utah in a big giant bubble wrap package. And then the science team at NASA will be the first to analyze those. But I collaborate with some of those folks, and I'm really hoping that I get my hands in a little bit of that.
And if you do,
I'm excited about sample return.
I am too, and we'll be talking to you many more times. I guess we're not returning these samples for 10 years or so, right? To 2031?
Yeah, that's right. It could be earlier than that, depending on how all these different pieces go and how funding goes.
So what are you going to do with these samples when presuming you get your fingers on them?
Well, the first thing I would do is collaborate with geologists, because I want to say the beauty of these missions is how interdisciplinary. Like, I can't look for biology in rocks without understanding the rocks.
Right, you can't get Perseverance to Mars without knowing the math. I know it's a separate discipline. Yes,
and it's the coolest thing, because so I'm, I collaborate with geologists so that I can understand how the geology and biology interface. So the first thing I would want to do is make sure the right people are on the project. And but what I personally would be interested in is looking for any signs of biology, and that might be DNA, but it might also be proteins or lipids or fats that only cells can make. And one of the things we found in the salt mine was cellulose. And you might say, well, I know what cellulose is. It's what
trees.
Paper, it's what paper's made out of, right? But cyanobacteria, these early organisms on Earth, actually made a ton of cellulose. And so to find that cellulose lasts for 250 million years on Earth, if cyanobacteria were on Mars and if they were making cellulose, I bet you anything that cellulose is there in the rock record. And on Earth, you can't see that molecule without it being made by biology, right? So we would know. We would know it was biology. I have no, I don't have it. I was going to show a stromatolite, but it's in the other room.
Sure. What is that?
A stromatolite is where cyanobacteria form a microbial mat and then cause the calcium carbonate to precipitate. Okay. So we see this on the bottom of Great Salt Lake. I have a Great Salt Lake one, but I don't have my ancient one here. So you can see the layers that are left behind in this rock. All of those were created by biology. So Perseverance is also going to be looking for these. They're going to be looking for layers in the rock that are shaped like microbial mats, more of a rounded shape because they were doing photosynthesis.
So show me that, show me that thing, the rounded areas that are not jagged, just those rounded areas, and that could not have been created by erosion or?
No. And when you look at the ancient ones, you see stripes that are in a mounded formation. And so what geologists say is, if sand grains were to fall, they have this, what they call angle of repose. They relax to a certain angle. If you see in the rock record something that looks like this, that's not an angle of repose. That was built by biology to get closer to the sun or the atmosphere. So that shape in the striations in the rock suggests that biology had something to do with it. So the oldest fossil record of life on our planet is 3.7 billion years old. And that's what it is. It's these stripes in the rock record that indicate cyanobacteria were making mats.
I want to ask you, and I want all of us, while you're talking, to pretend that this is Mars you're talking about. Okay. But what I want you to talk about is, what do you call it? The dissection of salt crystals and what you found in them, and how you, do you have a DNA laboratory at Westminster, or what exactly did you do to show ancient life here?
Oh, yes. So one of the coolest experiments I did was I collaborated with someone back at University of North Carolina called Jack Griffith, who is an electron microscopist. And we actually drilled into these fluid pockets inside the salt crystals. Do I have a salt crystal handy? I tell you, I had rocks handy and now I don't see them. They've all removed themselves. Yeah. So we drilled into one of these little fluid pockets and sterilely extracted the fluid and then immediately looked at it in the electron microscope.
How big is this fluid pocket?
So I measure everything in microliters because I'm a molecular type of biologist. But it about 100 microliters. So I would say that would be like just like a raindrop.
Oh, so it's rather large, I would say.
For a molecular biologist, yes. Yeah, it is.
And so inside these salt crystals that you find, presumably on the north side of the Great Salt Lake, there's actually little drops that that are ancient, that have been trapped in there forever.
So in the modern salt crystals, no, but in older salt, yes. So they are the age of the salt if they haven't been disturbed. So there's salt deposits, for example, at Death Valley that are 30,000 years old. And you can shine a microscope on those fluid pockets and see cells swimming around from 30,000 years ago.
What are they eating?
Well, that's an excellent question. And so one set of experiments that we are working towards is trying to understand the dormancy, like what genes do they turn off and turn on when they're in that pocket to maintain status quo, right? Because we think they go into some kind of sleep, right? Some kind of dormancy. Like think about a brine shrimp cyst. Those guys can hang out for a couple hundred years and then be resuscitated. And they're fully formed embryos inside those dry cysts, right?
I'm thinking of Horton Hears a Who.
Yeah.
Remember that? You know, where the elephant, exactly a little speck of dust. There was a whole society. Apparently, that's kind of what you're talking about.
Yeah, absolutely. So I think back to your original question, what I would like to do with those samples is do the same kinds of things we've been practicing at Great Salt Lake and we've been practicing on Earth, is to look inside those fluid pockets and to see if we can find evidence of the molecules of life. Right? Because we know what they look like if they look like Earth life.
NASA says, is that talking, the NASA press releases, and I love NASA, but the press releases are coming out and they're talking about in this ancient lake bed, clays, carbonates, and hydrated silica. That hydrated silica sounds like sand with water in it, hydrated silica. And carbonate the carbon sounds like life. When you see the word, what's that? Oh, that's it.
This is carbonate, calcium carbonate from Great Salt Lake. So the oolitic sand at Great Salt Lake is all calcium carbonate. We have a lake that is a calcium carbonate lake bed. And that's one of the things that is similar to this spot on Mars.
They say nothing lives in the Great Salt Lake, but that's not true because the brine flies, you know, they have a very short lifespan. They lay the eggs, they go to the bottom, they develop some air, they float to the top, the brine flies hatch, they mate, and the whole thing goes over and over again. And then we have bacteria. Are there several kinds of bacteria? And is that kind of what we're thinking about in Mars too?
Yeah, we've actually found about, I think, 56 different species of bacteria and archaea that we've found genetic evidence of in the lake. 24 species of fungi. I'm trying to remember of algae. I think we've got like 6 species of algae that we know for sure that aren't in this, that are eukaryotic algae, not that cyanobacterial kind, several different cyanobacteria. Anyway, it's very rich. If you were to shrink yourself down to the size of a microbe, you would be in a giant community where everybody had cool jobs and there were all kinds of different folks around. So the microbial community is very complex in Great Salt Lake, actually. We tend to think about the food chain as being very simple. Birds eat brine shrimp and brine flies, you know? But it's actually really complicated and in the microbe level, and those microbes turn over the nutrients, provide, primary productivity from the sun. They do photosynthesis and bring energy to the lake. They're really important. And I've spent my entire 2 decades and more here studying the microbes of this lake, not realizing until recently how similar they might be to places on Mars.
Okay, but I want to take a little turn here because all the sun that these, just to the Great Salt Lake, and some of your research, and I'm sure it has to do with Mars, but I want to take a little turn. Number one, into how sun is just decomposes everything. I don't know, decompose is the wrong word, but it seems to break down everything one way. But these microbes continue to live. I have read some of your stuff where you're looking at that as a way to help us as humans have better sunscreen.
Yes. So if you've ever been to Spiral Jetty, to that part of the lake that is the north arm that's cut off from the railroad causeway, it doesn't get fresh water. And so it gets saltier and saltier and saltier. And it turns out that on our planet, anywhere where we have water that gets hypersaturated with salt like that. So it's 10 times saltier than the ocean there. The ocean is like a little more than 3% salty, and it's 34% salty in the summer there. So it's
the Great Salt Lake is 10, right? The rest of the south end is like 10 or something.
Yeah, right now it's about 14 in places.
Okay, all right, but it's much 30. It's much
because
in the north end,
it's drying out. Yeah. So that pink water, you also see that pink water in places where humans make salt around the world in salt ponds. Like if you fly over Great Salt Lake and you see those beautiful patchwork colored salt ponds, you also see that pink color in Australia and salt lakes. So this is something that indicates the microbes there have pigments. And they do because they live in these harsh, dehydrating conditions with all this UV light. And it turns out that those pigments help them shield from the UV light and all of the kind of damage that happens in the cells. And so some of my research has been on those pigments and wondering about their effectiveness in a biotech proposition of, could we use them for sunscreens? So yeah, I'm super interested in pigmentation and sunscreens and how that might help people.
Any progress in your research?
With respect to defining what those pigments do, yes.
Okay.
But nothing with respect to marketing that for humans.
All right. Well, hurry up because sunscreen doesn't last long. Hurry up. Okay. Now the other thing you brought up these two subjects, and I want to explore the other one, and that is the food chain, where the birds, eat the brine flies, and the brine flies. What do they eat? The shrimp or the bacteria? I don't know what they. But anyway, we're talking about pollution. A lot of people say, you know what, it's the Great Salt Lake. It is what do they call it? A terminal lake, or because it doesn't go anywhere?
Yeah, it's a terminal lake because it's the bottom of a basin. So the water doesn't go to the ocean.
So who cares about pollution? And your answer to that with regard to the food chain?
Well, okay. My first answer to that is that we have 10 million avian visitors a year, 338 species. And those birds, by definition, as migratory birds, they migrate, right? So you might not care that there's mercury in Great Salt Lake.
No, I care greatly. I'm just putting it to you.
If the brine fly larvae are eating that, and ducks are eating the brine fly larvae, diving and eating, and then flying somewhere else, they're taking that mercury with them. And so I actually went to testify at the EPA 2 years ago on these mercury rollback propositions because, and there was a representative coming from every state, and I was the only one who talked about mercury in ducks. All of them were from Trouts Unlimited. They were talking about mercury in fish. So we have a really unique mercury problem in terms of getting mercury directly into ducks that humans eat. And most people, again, the US Clean Water Act really only has to do with water that fish live in. So Great Salt Lake doesn't fall under that. And our good people at the state agencies in Utah have tried to come up with standards to help us keep Great Salt Lake clean, but it is super important. And because it wasn't drinking water, it wasn't thought of as being a water body that needed to be protected.
Do you know where this mercury comes from?
Well, Utah likes to blame all their problems on Nevada, right?
Okay.
So,
okay.
It is elemental mercury is a natural byproduct of gold mining, gold smelting. And so then it would come over from Nevada as an atmospheric mercury cloud and meet with chloride from Great Salt Lake and become soluble. So We think that's one possible avenue, but burning coal also, I mean, back east where, mercury contamination of water came from all the coal burning. And once people stopped burning coal back east, then they got rid of that. So it's certainly coming from atmospheric deposition, but it's not easy to say exactly where that comes from.
I appreciate.
We do know the microbes in our lake are turning it over and making it more toxic. So as the lake recedes, the other answer to why we ought to care is about air quality, because that salt dust is going to blow into the Wasatch Front. And
that's right.
We're going to be breathing that mercury. So
because it gets windy, the lake goes down, it gets windy, and we see all that dust come up, and that's what it is.
That's right. That's right. So we really ought to care about heavy metals in the lake.
You have we've gone way off topic here, and I appreciate it, giving me the whole lecture. But are you going to show us a Martian one day to get back to?
So. I'm there are, I find in this kind of NASA work, there are optimists and pessimists, and I'm definitely in the optimist camp. But I do know that Mars went through some catastrophic changes a long, long time ago. So it's a big ask. Once the surface water was evaporated 3.5 billion years ago, it's a big ask for something to lie around that long in the rocks. I mean, 250 million years on Earth is a big deal, but 3.5 billion is a different number. So I'm hopeful that we at least find signs of biology. And if it is similar to what we have on Earth, that then we can start to build relationships and think about, did the two planets share material over time and exchange microorganisms early in their history? So I think these are super interesting questions.
They really are. And I am so grateful that you agreed to come on and talk to us a little bit about this. And I want to stay in touch, because something is going to happen between now and the next 10 years, and you're going to know about it. And I want to know about it. I want our whole audience to know about it. Bonnie Baxter, Dr. Bonnie Baxter. Yeah. Thank you very much for joining us.
Okay, cheers. Thank you so much.